I'm taking a slightly different approach, and I want to show how the compute facility that's centrally accessible has really allowed the research that I do to flourish, and without it, it just could not be at the scale it is now. And so I'm going to talk you through what that research is and why I think it's important and exactly how it's starting to grow now. So my group looks at microbiomes in the environment. We want to try and understand how we can engineer those microbiomes to solve environmental problems. Underpinning that is microbiome science, which is computationally very demanding work. And it's one of many moving parts that you need to be locked in place to be able to do any of this. So although our interests are environmental, there are massive repercussions, positive benefits to be had in health and soil and all the other areas where microbiome science is really important. And why is that? Well, there are three really good reasons why microbiomes are so important. Isolation is not a natural state for microbiology. So although microbiology up till now has looked at studying individuals and isolating them, understanding how they operate on their own, that really does not reflect the natural world at all. And partly because of that, we have only been looking at a very small proportion of diversity of life on Earth. So the majority of microorganisms can't be isolated, so we don't know what they do, unless we apply very fairly modern, very powerful techniques that rely on genomics. And this is an updated tree of life. If you can imagine, only 30, 40 years ago, there were only a few branches on that. But now we've got all this, what we call foliage, and this has all been uncovered using genomic science. It's now very cheap. Getting the data is easy. Doing something with the data is not. I'll come back to that. So we now understand there's this enormous diversity of life on Earth that we really don't understand very well. And we also appreciate that it works together. If you understand one organism in that, it doesn't really tell you what it does in the natural world and how you can harness it. And this opens up a really exciting biotechnology potential, because if there's a huge amount more diversity of life on Earth that carry out a much more diverse array of functions, many of those are probably useful to us. And if we can understand them better, then we can try and harness microbiomes rather than the individual organisms and strive for more efficient, cost-effective, more successful biotechnology interventions, as well as just understanding the microbial world, which we really only have a very narrow view of now. So my group focuses on carbon cycling. Carbon cycling is fundamental to all life on Earth, probably life beyond Earth as well. That's a conversation for another day. And it has an impact on all the other globally important biogeochemical cycles. But it's also where our biggest environmental problems are. So two good examples are carbon dioxide emissions, which are the biggest driver for climate change, and plastic pollution as well. And both of these problems could be more efficiently tackled, I believe, using microbiome engineering. So understanding what microbes do with carbon is what we're trying to do in order to try and harness those processes to better solve these problems amongst others. So how do we actually do that? Well, it's enormously complex and quite labour intensive. So underpinning everything we do is this whole process which we call metagenomics. So rather than just trying to understand the genomic content of one organism, we take a community of organisms, sequence the whole lot, and try to make sense of that using computational tools. And there are so many rich data sets that you can mine and huge insights that you can gain from doing this. There's a non-exhaustive list to the right that you can really delve into answering some of those really fundamental, important questions using genomic data alone. But these are big data sets. So you can sequence the DNA very easily now and pretty cheaply as well. And this is a very, you don't need to worry about the detail here, but this is an example of the type of workflow that you need to then apply to your raw data. And I found this nice paper earlier, which is sort of 10 simple rules for investigating metagenomic data from environmental ecosystems. It is a bit simpler if you're looking at human ecosystems or animal ecosystems, because they're less diverse and we know what they look like already. But in rule 5 is, you need massive compute resource. And the type of compute resource you need is quite different to what the mainstay of the CSF and our central compute system has had over the years. So it's very RAM intensive, and it needs a large number of high memory nodes spread across lots of CPUs, not GPUs, which is what spatial modelers tend to rely on. This is a ever so slightly unfinished slide, but basically I wanted to profile two uses that we have that have allowed us to operate basically and continuing to allow us to grow. So we have our own group server, which is part of the Edge Compute facility, and that's managed through Research IT. That is 1.5 terabyte RAM node, which, believe it or not, is way too small. And it comes with about 12 terabytes of storage, which is at capacity. So we're talking, and that's not, that's one project or two projects. And we have a direct link to the computationally shared facility, which over the years, and really it's been over about 5 years now, the repertoire of different software tools that we rely on has been building up and building up, so that lots of people can now access the CSF and the software and these high memory nodes that have been added, many of them directly because of the Research Lifecycle Programme. So this is an underpinning resource that enables us to do pretty much everything that we do now, or the metagenomics aspect of it. And this is a hideously complex example of just running 3 samples through an automated pipeline, that many steps of which require very large amounts of RAM. Not all of them are computationally intensive, but the key ones really are. And so this is the kind of approach we're now able to apply to huge numbers of data sets and automate that analysis. So to put a spotlight on one example, we're doing currently a global survey of the metagenomes that have come from hot springs, because we want to try and engineer communities of microorganisms to convert waste CO2 into useful products. And we want to understand what they can do before we start trying to play around with them. So a postdoc in my group, Chris Stead, has collected all the previously sequenced data sets, as well as added in a few of our own. There's about 70 odd samples, and he ran those through our pipeline, which I should mention was developed on our Edge Compute node, and then we can shunt that over to the CSF so that we can allow the bigger compute nodes to work its magic there, and there's this really nice connection there. And we can start doing some really quite large scale analyses of you don't need to worry about the detail, but this is taxonomic profiling of all of these 70 samples that we've now reanalyzed. So we know who's in what, and this is actually a temperature gradient from left to right, so it gets hotter as you go along. And really what we care about is understanding, and again, you don't need to look at the detail, but this is distilling what is actually a huge amount of data into some biological meaning by understanding what the potential pathways for carbon fixation are. And being able to do that globally allows us to really take a holistic view of that different environment, rather than just take a single snapshot of one sample, which you could do on a Mac, for example. You could do all of those steps on a Mac, but it would be without any kind of, there's no benchmark there. So all of this has been incredibly useful in allowing us to pull in an actually massive grant that puts, microbiome science on the map here at Manchester. And we would not have been able to convince the funding agency to give us this had we not already had pretty sophisticated and demonstrated compute facilities in place, because it's one of, like I say, many ingredients that you'd really need in place to do microbiome science. And then the flip side of that is we were able to cost into this grant another Edge Compute facility, which will be a 4 terabyte RAM node that we can then do a lot more dedicated stuff on, that will eventually migrate over to the CSF and allow more users to leverage. So for that particular project, we're really trying to do something like this, which is, can we turn the extremophile communities in hot springs into factories that can convert CO2 into useful things? And this is a very complicated, summary of a very complicated 5-year grant. But the point here is that, my interests are central to sort of net zero, but this can also be leveraged, this kind of science, to try and strive for a circular economy. I mentioned plastic degradation, and polymer degradation is something else we're doing, collaboration with the likes of Unilever. But then it also has really important implications for health, for gut microbiota, skin microbiota, and then sustainable agriculture as well. So it's really an underpinning technology, and it's so heavily reliant on having good computational resources that are actually very hard to access. So I think that's something that we at Manchester are very lucky to have, and it will continue to grow. So thank you for your attention.